Hi, welcome to the Cell Biology Laboratory at California Lutheran University. Today we're going to do a demonstration of mammalian cell culture. Cell or tissue culture can be accomplished by growing cells in sterile dishes with a nutrient-rich liquid media. There are two basic types of cells that can be grown in culture: adherent cells, which stick to the bottom of the dish, and non-adherent cells that will float in the media. Caution must be taken when growing non-adherent cells to not throw away your cells when discarding old media. First, let's take a quick look at the materials that will be needed for cell culture. So the first material that you'll need are gloves, 70% ethanol in a squirt or spray bottle to disinfect areas, a cell culture flask with a screw top. Alternatively, you can use cell culture dishes that have a loose top. Each one of these flasks or dishes can come in multiple sizes for how many cells you need to grow at a time. Lab tape for marking tubes and other equipment. A lab marker, preferably one that doesn't dissolve upon ethanol application. 50 milliliter conical tubes for holding media, tube racks to hold your tubes, and 15 milliliter tubes for holding cells or media with cells in them. Also, you'll need a waste beaker for discarded media and pipette tips. And most importantly, you'll need serological pipettes. These can be plastic or glass and come in a range of sizes. This one happens to be 10 milliliters and will hold up to 10 milliliters. It has divisions in 0.1 milliliter increments. Here's a 5 ml. Here's a 2 ml. So depending on the application, choose the right volume of serological pipette. Also, you need some paper towels to disinfect surfaces. So any manipulations with mammalian cells and culture should be done inside a biosafety cabinet, otherwise called a tissue culture hood. This provides a sterile flow of air over your work area and prevents any sort of contamination, bacteria, fungi, any mold spores from getting into your cultures. These things can outgrow your mammalian cells and contaminate your cultures and eat up all of the nutrients in your media. So to work with a tissue culture hood, first switch on the blower and then switch on the UV light. While the UV light is on, it is a mutagen, so you'll need to stay out of the area and don't expose skin to the UV light. After the hood has been decontaminated with the UV light for 30 minutes, switch off the germicidal light and switch on the fluorescent light for working. At this point, you're going to want to decontaminate the surfaces with any particles or still living bacteria or viruses. Spray all surfaces down with ethanol and wipe clean with a paper towel. When doing tissue culture, you'll need to use a pipette controller like this one with your serological pipettes. It has an up and down buttons to suction up liquid into the serological pipette and to dispense it. Turn the power on at the base, and on your serological pipette, open the packaging to keep the inside of the packaging and the serological pipette sterile. Put the end of the pipette into the pipette controller, make sure it makes a good seal, and slide the wrapper off without touching the lower end of the pipette. Keep the lower end of the pipette sterile so that anything that goes in and touches liquid, media or cells, stays sterile. Draw the media up with the up button and dispense the liquid with the down button. Once you're finished using a pipette, remove it from the pipette controller and throw it away. The next step is to prepare the tissue culture hood and all your surfaces and equipment so that it's decontaminated. Spray ethanol on your gloves and rub, covering every surface to make sure you kill any existing bacteria and mold spores, fungal spores. Also spray equipment like tube racks and any tubes containing media. There's no need to wipe off the ethanol, it will evaporate quickly. You do want to be sure not to spray anything that actually has cell culture in it, as the ethanol vapor could kill your cells. There are several considerations when using the tissue culture hood. Don't block any of the vents with the pipette controller, paper towels, or other equipment, or your arms. Also, try to avoid setting anything down on the surface that may touch your cultures, such as lids. Instead, unscrew your cap and gently set it back on top of your tube. Don't block any of the back vents either. Try not to make the tissue culture hood a storage facility by stacking things in the back, covering up those vents. The next step in cell culture is to prepare a complete media that has all the nutrients needed for your cells to survive. The first ingredient is a basal media. The basal media has salts, sugars, and certain other elements that are needed by the cells. They come in different formats, and this one happens to be DMEM for Delbecco's minimum essential media. The media is red because it has phenol red in it, a pH indicator. The next solution would be fetal bovine serum. This has many growth factors and proteins needed by the cells. Also, penicillin and streptomycin are needed to reduce bacterial growth in your cultures that may contaminate them. Lastly, L-glutamine is an amino acid that can degrade rapidly. This must be added to media in order for the cells to survive. The complete media I've described here is sufficient for many types of cells, but every cell type has its own requirements. Protocols may ask for additional additives to the complete media, such as growth factors, other amino acids, or things like beta-mercaptoethanol. Consult the literature for the right additives for your cell type. Complete media is usually made in concentrations with serum at 10% to the final volume, penicillin streptomycin at 1% to the final volume, and L-glutamine at 1% to the final volume. The rest of the volume would be made up by the basal media. The most important piece of equipment in doing cell culture is a microscope, specifically an inverted microscope where the objectives are below and looking up through the specimen. This allows us to look at cells that are growing on the bottom side of a flask. By placing the flask on the stage, you can then see the cells that are growing in the liquid media. Use the focus knobs and look through the oculars. Cells are grown in a water-jacketed incubator. Open up the outer door in order to see the cells growing in the inside. There's also an inner glass door, and inside the chamber, the temperature is 37 degrees Celsius, body temperature, and 5% CO2, much like the hypoxic nature of tissues. Remove cells from the incubator, being careful not to spill or splash the media, and look at them under a microscope. To feed an existing cell culture, the first step is to use a serological pipette to remove the old media that's been consumed by the cells. Using sterile technique, pipette off the old media, being sure not to scrape the bottom of the dish where the cells are growing. Discard the old media in a waste beaker. Open the tube containing your new complete media. For a 10 centimeter dish, pipette between 8 and 10 mils of new complete media onto your cells, making sure that you either pipette down the inside rim of the dish or gently onto the cells, being sure not to displace the cells by pipetting too forcefully. Rock the dish gently to make sure the media contacts all the cells, and throw away your serological pipette. When splitting cells, first remove the old media and wash with a serum-free media. Next, add 1 to 2 mils of trypsin to your cell culture in order to digest away the proteins holding the cells to the bottom of the dish. The trypsin usually takes about 5 minutes to work and dislodge the cells from the bottom of the dish. At this point, return the dish to the incubator. Once you can see the cells dislodging from the bottom of the dish through the microscope, add a media containing serum, sometimes called a quench. This is to stop the trypsin reaction from eating away any more of the proteins on the outside of the cells. At this point, you'll want to transfer the media containing cells and trypsin into a 15 ml tube, which can then be centrifuged to pellet out your cells. Once you've transferred your cells into a 15 ml tube, centrifuge the cells to the bottom of the tube using a centrifuge. Open the magnetic lock door, place the cells in, and make sure that you've balanced the rotor on both sides. Press Show Set, Enter, and start the run. Centrifuge the cells for 5 minutes at 1800 RPM. Once the cells have spun down, bring them back to the tissue culture hood and decant the supernatant. Flick the pellet between your fingers to make sure you dislodge the cells from the bottom of the tube. Add new media back to your cells and pipette up and down to resuspend the cells in the new media. At this point, distribute the cells into your new dishes containing new media. The volume that you resuspend the cells in and the volume of media that you have in your new dishes is completely up to you. Label your dishes with the cell type, the date, and your name, and place back in the incubator.